So this is the last session before lunch, and Guy and Ellie are going to go through what we are going to need to do to prepare for our clinical trial readiness. So it's very important. You're almost at lunch, guys, so please pay attention for this last session. Yeah, and actually, the work that's going to follow what we have done is going to be called a standard of care. There is no standard of care for FOXG1 right now. And Ellie's like crazy, she does the most crazy graphs that I don't even understand half her graphs, but the one about the epilepsy journey, what we found, and I thought was shocking, is that there was no two children with FOXG1 who have epilepsy who have the same medication journey. I mean, it's nuts. It's absolutely insane. So there is no standard of care. And that's why this work is so important, so we can define that to answer those questions. Absolutely. Yeah, that's right. Okay, we are up on Zoom, so hopefully everyone's relogged back in, and I'll hand it over to you two.
Alrighty. So the list of things that scare me isn't very long, but one of the components there is being in between a hangry audience and lunch. So we'll try and do good. So this I think this panel, if you will, is less structured, right? We don't have a slide deck. We don't have a specific sequence. We have 2 slides, but it really is more of a sort of free conversation. And the purpose of this is, like, the timing is not, it is not accidental. It's after you got an overview of kind of the path that we're on, where are we along the path, and what did we learn with your help about FOXG1 and the symptoms and the areas that matter. And in a way, it all culminates at the end with the last slide in Elli's and Kapika's presentation in terms of the key domains that with your help we know that as we design a clinical trial we need to hit: the communication, the movement disorder, right, the sleep, all these domains that we need to address, because those are the domains that matter. So just identifying these domains is critical. It's absolutely essential. We need to measure the right things. Now, the next challenge is, we know domains-wise what we need to measure. How do we measure them? What are the scales? What are the tools? So, let's start with a hypothetical. Let's say that we develop a new drug for high blood pressure. Drug development isn't easy, but the path of, like, how do we approach the design of a blood pressure lowering drug is paved, because we know what a normal blood pressure range is. We know what an abnormal blood pressure range is. So we know who we're enrolling to the trial, and we also know how to measure the drug's performance. We can define success quite easily, and we can define failure as well. So if my blood pressure is 160, and after I treat for a few weeks it's 159, then it's probably not great, right? This is probably not a drug that works. If it's now 125, okay, that looks pretty good. How do we do this? Well, we enroll patients and we give some drug, and others are going to be in placebo, right? So we have a control group so we can compare, so there's no bias. And we compare the performance to a control group, and the expectation is that a control group will not improve, and hopefully the group on the drug will improve. So we know how to define success. We know how to measure it. We have a scale for that. Let's say that we develop a treatment now for Parkinson's disease. There is a scale, a rating scale for Parkinson's disease. If we develop a depression drug, there is a known scale for a depression drug. So our success or failure depends on if our drug is good or not, but the path to test it, measure it, and declare failure or success are known. There's no FOXG1 rating scale. It doesn't exist. So when we think about designing a trial and testing for treatment effect of a drug, there's a trivial, non-trivial question of how do we actually measure it? So we know that we need to measure now communication and movement disorder, and sleep, and et cetera, but with what? What are the tools? And what are the most appropriate tools to do so for FOXG1? Because there are measuring tools out there, but nobody has experience with this. To the best of our knowledge, there has not been a clinical trial, disease-modifying clinical trial for FOXG1 syndrome. So I actually don't know how these various scales out there perform for FOXG1. Are they even relevant? How long do we treat in order to measure a treatment effect? What is a meaningful movement on a certain scale? Do we need to move it 1 point or 3 points or 30 points? What is success? All these are critical in order to sort of design a proper informative clinical trial. So this is our problem statement, right? We can add another layer or 2 or 5 to this. One of them, for example, is given the procedure for administration, et cetera, we probably don't want placebo, right? We want to treat. And so we want to treat all the patients in the trial. One of the conversations that we're going to have with FDA and the regulatory agencies in other countries is whether we're going to be required to do placebo or not. We don't want to do placebo. But here's the catch, okay? You saw Elli's data, you saw Kopika's data. Our patients, our kids, they develop slowly. They plateau at some point, but there is some development. So if we don't have a control group, like with the blood pressure trial, then we can treat and then we'll see some improvement. But let's say that we treat a 2 or 3 or 4-year-old child when they're still developing slowly, and we don't have a control group, and then we'll see some improvement. How do we know? How does FDA know that this is due to the drug? Or if you haven't done anything, you would probably still see that certain improvement. So if we don't use a control, how do we assess whether we are really effectively treating or not? So those are some of the challenges that are unique to FOXG1, but not just to FOXG1, to rare diseases like FOXG1 in general. Elli, you're enjoying?
But actually, would it be possible to have the slides put up?
Okay, so we have one.
Oh, amazing. Thank you.
Great. So this is where we need your help, right? Once again, not for the first time, not for the last time, but this is where we need to partner with you. Because through conversations and an incredible partnership with our clinicians, right? We have Dr. Benke there in the back and others. We are working through and figuring out and brainstorming and planning what are the most appropriate outcome measures, scales, tools to assess treatment effect in a clinical trial. And I see a question there in the back. So there are different ways of designing a trial, but one of the things that we're trying to avoid is actually avoid using a control group such that all patients on the trial are going to be on treatment. And so, in one of the challenges and one of the what's so important about the natural history study, and what we're going to ask for your help with now is to help us gather information that would strengthen our ability to dismiss the need for a control group, such that all the patients can be on drug in a trial without placebo control, but still to enable us to make, to properly assess the treatment effect of the drug itself. Okay. It's complex. I think it's doable, right? We need to do the right things ahead of time in order to enable that. And part of that is to continue and deepen our understanding of how our patients, our kids perform on specific scales. So now that we have, and this is like a brilliant slide that Elli prepared, where of sort of the path, right, of how we go there. And so, Elli, maybe you want to take us through the steps here.
Sure. And to be clear, I recently discovered an AI slide maker tool, so this was not me. So we've talked a lot about where we are today, which again is in this preclinical state, and we want to make the jump into clinical. And so what we've been focusing on historically is how do we define what we're going to measure? And so those of you may remember probably like 5 or 6 years ago when we launched the patient registry, that was really step one and continues to be one of the most important mechanisms we have to collect data about the global FOXG1 syndrome population. We have our natural history study that's complemented others that are ongoing, as well as the disease concept model study that Kopika talked about today. But what Gai is going to be introducing here is, now that we've again identified what we want to measure, we need to establish how we're going to measure it. And to Gai's earlier point, if we want to propose to the agency that we won't have a placebo arm in this trial, meaning that every participant would receive drug, we need to have a really strong rationale that we understand what the baseline looks like so that we will be able to identify change if and when it happens. And so that leads us into kind of phase 2 of our journey.
Does that make sense? Leah, I know you asked the question on the placebo.
That is correct. Now, the ton of data that we have, there's one thing that we don't have there. We have so much stuff, but there's one thing that we don't, and that is that the scales that will be used in a clinical trial are not necessarily the scales that are used in everyday clinical practice. And so while we have a good sense of the symptomatology of FOXG1, with time, what we don't have is how our patients, how our kids perform on specific scales that we think are the most likely to be informative in the context of a clinical trial. And this is where the call for action comes from us, right? So we are now working with our partners, with our clinicians, with other foundations, right, that are in the same stage as we are, and we are collaborating with them in order to establish a certain baseline of how our kids, FOXG1 kids, are performing on the specific tasks that we narrow down as our, like, the top of the list of assessments that we want to use in the clinic. Now, if we just go blind into the clinic with them without having any establishment of baseline on how we perform, then we're crippling ourselves, right? So the more we know about how we perform, how our kids perform on these specific scales ahead of time, the better we can design the trial. And the more, just the stronger the case will be. Now, if we, it's not just baseline. Let's say that, so what we will ask, we'll ask your help with is to, once we roll this out, it's not ready to go yet, but once we roll this out, which we hope will happen in the coming months, is to help us by using those scales, those assessments. Some are going to be clinician-based, so we may ask you to go to the clinic and meet a physician that'll do it. Some are going to be assessments that are questionnaires for you, right? And what we'll want is to ask you, say, to measure something or assess something at this point in time, and then maybe 6 months later, and maybe 12 months later, such that we have also some longitudinal, right? We see a certain trajectory. So if we measure now, and 6 months from now, and 12 months from now, is it the same? Is it better? Is it worse? There's no treatment, right? We're just establishing the performance on specific scales over time. This helps us establish the baseline. This could help us when we engage with the agency and we justify why we don't need a placebo, because a baseline measurement of a performance is a good enough control to compare a treatment effect to. We actually need data, right? And we can't get it without you. So one effort that we will enroll soon is the effort to gather data on how the kids perform on outcome measures, on specific outcome measures, scales that will be used in the trial. A second effort is the digital biomarkers, which we're partnering hopefully with another foundation. And the key there is, because we have sleep issues, we have movement disorder issues, there are some devices, like you can put a device in the room that is a sensor that assesses sleep, it just assesses movement, right? And we can quantify how much movement, how much disruption, how much quiet time of sleep there is at night, establish that as a baseline. Another is something that is like a Fitbit or an iWatch or something like that we can put either on the wrist or on the ankle and measures movement. Another way to get information, data that later could potentially be used in the clinical trial to show improvement on the movement disorder. All this requires us to learn how these measures perform before we decide if and how to implement them in a clinical trial. No. Nothing. Okay. So really, I think that this is really the message. This is the purpose. I think
Yeah, let me summarize
here.
this message, okay? It's going to be a little bit of a pain. We're going to have to do a few things, but we all have to do it. And that's it, because otherwise we don't have a drug. It's that simple. And our goal is to try to minimize that pain. We are having lots of debates with our clinicians. We're having debates with the agency. And when we say the agency, we don't just mean the FDA. We're also talking about European and other agencies. We're having debates to minimize patient burden. That's why we created the digital natural history study to begin with. We understand patient burden, but there are going to be a couple things that we ask you to do, and we just all have to do it. And trust me, it's a pain for me too, but I'll be doing it. So let's just all get together and get to the finish line.
Nasha, you don't need us. Just say that, and we're done, right?
No, it's so nice that you explained it, though, why we needed it. But there was a method to our madness, why we created these different things, why we've been asking you to join interviews and do this work. It is because we have been working with a strategy to get to the clinical trial. So just so y'all know. Okay, questions? Yeah, go ahead. We'll get a mic to you. Get 1 sec. Okay, go for it. Oh, sorry. No, it's for the virtual. It's for the guys on Zoom, too. Just give 1 second.
So when you say you need all of us, I think it's good for all of us to hear what you mean. You need 100 of us, not 10 of us.
That's right.
Right? So I think that's a really important message, because I can see from looking at the natural history study and the disease concept model, the number of people who found some way to do it. So I think that's a takeaway. Mm-hmm. Everyone's life is busy. Everyone has someone screaming in the background. So this has to be a priority over all those other things that are screaming at you to do. It can't just be a few of us. It has to be every single one of us.
Exactly. And, thank you for saying that. We have about 500 patients in the United States. We only have the natural history data on 100. look, I'm so thankful for those 100 patients, because without that, we would not have this study, and that we're going to use that in our filing to these agencies. But we could have had 500. It would have been a richer study. We would have understood our community and these symptoms more. So I cannot echo that enough, that every child's participation is so critical when you have such a small disease. If this was breast cancer, you want to participate, do it. You don't, it's fine. There's millions of others who are going to participate. We don't have that luxury. We all have to participate. So thank you. I think there's a question back here, right?
Is there a target age range for the clinical study? In the patient age, the younger Fox kids, or, because I saw you broke it down with the under 15 and over 15. I didn't know where the target would be for the patient.
So the target will need to be determined through a conversation with the agencies, right? Because they have a say there. From our point of view, the goal is to have the trial be as inclusive as possible across the various age ranges, such that if it's successful and it gets approved, it is approved for the broader patient population as much as possible. But the actual age ranges it'll have to be a dialogue with the agencies.
Right over.
For establishing the baseline and the definition of the baseline, there are definitely many organizations dealing with rare diseases in the world. Is there anything that, for instance, we from Europe, how we can contribute to talking to such organizations to find out what they have already done, not to reinvent the wheel?
Yeah, we are absolutely doing that. And when Guy was mentioning, we're working closely with the Rett Syndrome Research Foundation, for example, who are global, and they're in 2 clinical trials now. So some of the biomarker work we're going to do will be in partnership with them. We're also working with the Angelman Foundation on some of their scales. So absolutely, we don't want to reinvent the wheel. We're trying to learn from others. And we're actually trying very hard to not create a specialized FOXG1 scale, which then becomes more difficult for the agencies, takes longer for drug development. We're trying to actually piggyback what's already known. So yes, 100%.
Yeah, and I'll add here, this is one of the advantages of being a nonprofit patient group, is that we're non-competitive, right? Yeah. It's not one company competing with another company to win over a market. It's not the case. So we can partner with and we are partnering. Right. We're learning from others are learning from us. It is an ecosystem of non-for-profit organizations that work together, and that's a huge advantage.
Oh, right.
I think
do you have a...
Oh, sorry, just one follow-up just for the international folks in the room today too. I think just something else that could be helpful to consider and flag to us is something that maybe this is not a surprise to you, but it was a surprise to me, but something that actually takes the longest amount of time in setting up studies like these is the coordination, the contracting, like the data sharing agreements, all of the bureaucratic stuff that you don't want to be spending a lot of time and resourcing on, but something that can really accelerate that is if you have a real champion. So if you're physician or a scientist in your country that is in your corner and understands the urgency, definitely identify those individuals to us, because I think that could make it a lot easier for us to establish some of these sites in other countries as well.
Yeah, exactly. In fact, Steve in Australia has done a great job working with clinicians and, getting a center together. So if we can do a clinical trial in Australia, we would be able to do that. And so, yes, to Elli's point, if you are international and you have a great neurologist, we'd love to hear from you. I know there's online.
Yeah, just a couple quick ones from our virtual attendees. One is outside of the U.S., how can we help with this? Sounds like we've answered that. The other is, where can I send my son's information, medical information, to help with these trials? So how do they actually operationally do this?
Okay, great question. So if you go to our website, FOXG1research.org, and you go under Parents and Caregivers, if you're anywhere in the world, we have the patient data center. And so if you haven't registered your child there, please do that. There's basic information. We ask you to upload your genetic report, and there's like a couple surveys. So that's the first thing you should do. If you're in the United States, then also please go, there's a link for the natural history study on Citizen. Joining that takes 5 minutes. We've tested it. It does not take more than 5 minutes. So all you have to do is go online, you put in where your child has been seen, and then we will do the rest of the work to go collect all of your children's medical records. Please know we are under IRB, so it's the what does it stand for again?
Yeah.
Thank you, Institutional Review Board. And so the data is protected. We cannot share it with researchers unless it's de-identified. So there's a lot of care we take on that. And if you have questions, we're happy to answer those questions. But those are the two things that you could do.
Nasha, do you want to speak to international participants?
Yeah, and so international, again, you could go to the patient registry, and we will be expanding the Citizen platform. If Elli could work faster, it would be great. So we're doing a couple pilots around the world to figure out how to get that rolling, but you will be able to participate. And then in the work that Gai is talking about with the establishing baseline, we are going to make that available internationally, and so stay tuned as we come through with that. And then I think, Kate, did you have a question? Gone? Yeah. Exactly. And so, why the work Stephanie and Monica and others are doing in different countries is so important, because just gathering the patients is critical, not just for the clinical trial, but when the drug is actually approved, having a database so we can even let patients know that this drug exists is going to be critical. So the country leads are doing a great job. In the United States, we've broken that down by a region. And if you would like to get involved, there's folks in different states that do get-togethers to get the community together to understand who patients are. We need more volunteers, but a big part of that is just figuring out who is where and a way to contact them.
I wanted to ask, is the rescue of the corpus callosum maybe a possible measurable outcome during the clinical trials?
So this is something, so the actual design itself is still ongoing. There is a lot of variability in that. So there's a question of to what degree that relatively small number of patients that will be enrolled, what's the phenotype there? And it's also not quite clear what will be the relationship between any changes in corpus callosum as a predictor of treatment effect. So it's a possibility, but the priority will be on the functional outcomes. So this could be another imaging exploratory endpoint, but generally speaking, if we see any correction of the corpus callosum in patients without a functional treatment effect, that it's not good enough. And if we see a functional treatment effect, then what happens with the corpus callosum doesn't matter. And so it is in the preclinical sort of space, it is actually important because mice recapitulate certain elements of FOXG1 syndrome, but they don't have FOXG1 syndrome, right? And so those histopathological or anatomical abnormalities in the brain, which are corrected, which is amazing, those are the best surrogate that we have for a treatment effect. Whether or not it predicts cognition and et cetera, all these functional aspects that a mouse can't always tell us, right? This is where the clinical trial comes in.
Peggy, I think the only thing I would add to that is when you look at how the agencies are defining endpoints, it's how someone feels, functions, and survives. And so something that gets tricky about a specific anatomical anomaly that, in the case of corpus callosum anomalies, are not always associated with an outward phenotype or difference in humans, at least. That one might not quite make the bar. And part of why we've been really focusing on building this body of evidence is that the FDA wants to see that we've been really thoughtful about what we've chosen because it's important and meaningful and life-changing to families. So it's part of the considerations that we've had as well.
Yeah. And that's Peggy, by the way, who is a FOXG1 parent, obviously, but a pharmacologist and part of our clinical team and does a lot of work for us. So thank you, Peggy. Any other questions? Ryan, did we have any more online?
One last look.
Any other questions in the room?
Like we do have one online.
Okay, one online.
Ideally, how much dose required to cure? Does it vary from mutation to mutation? And then how a scale was designed for the SMA drug? Those are the two questions online.
So with the SMA, it's a bit different in that SMA is a lethal condition. So there are functional endpoints, but survival is really the key, right? So it's a bit different in that respect, and very thankfully for us.
What about for Angelman or Rett? Do you want to talk about maybe those endpoints?
So we're looking carefully at the endpoints that are being used for them, and what we're doing in the name of not wanting to reinvent the wheel and learn and adapt or adopt existing outcome measures to FOXG1 syndrome. Yes, we are. So those are the ones that are being used for other conditions are the ones that we're looking at carefully and are going, with your help, we hope, assess the suitability of them for measuring performance in FOXG1 syndrome. And then we'll assess whether we can use them as is or we need to make some adaptations to them, but then we can still use them as a template and make slight modifications, refinements if needed to optimize them for FOXG1.
So yeah, the other question was, ideally, what would the required dose be to establish a curative effect, and then does that vary by mutation?
So the dose selection will be, and we touched on this earlier, will be based on the safety, preclinical safety and efficacy. Those are the studies that are ongoing now to understand the safety margins of the therapy, of the drug, and how low can we go in the dose in mice and get a treatment effect. That defines a therapeutic window, and so we don't have the answer yet because those studies are ongoing. Once they complete, that's when we'll have our second meeting with FDA, and that will include a conversation and alignment on the doses that will be used in the clinic.
Thank you. Okay, one last question. Go ahead. No problem.
I hope my English will be enough to explain what I want to say. There is a lot of talk, at least in Europe, where we are from, about GLP-1 drug antagonist that is being tested for various kinds of, say, diseases. And it is diabetes, it is Alzheimer, cravings, addictions. And for me, diabetes has nothing to do with Alzheimer. Still, it is the same drug that is being tested in both cases. And with such, they call it in the articles, one drug that can be tested for many different diseases. Is it something that you could also use just to check if there is something from this drug that could be used in, and is there a way, because I have no clue, if there is a way to check if we can also contribute from what is already in the science regarding this GLP-1 drug antagonist?
You've tapped upon Guy's favorite topic, which is drug repurposing. He has very strong feelings about this. Go ahead, Guy.
Yeah, maybe we'll. That's not over lunch. That's over a drink, right? But
All right.
you got it. Yeah. So this is a fair and good question, and I think that it's not specifically for GLP-1, but more general around repurposing. There's a lot of work around this. There's something very attractive about the idea, in that there are already drugs out there and they could potentially be repurposed, especially if they're approved already for something, and then the regulatory path, the timeline for approving for another indication is definitely favorable. The downside is the likely lack of specificity, right? Because if it's working or presumed to be working for everything, then what exactly is it doing? And what is the mechanism that makes it relevant for, say, FOXG1? So the foundation has finite resources. And we had plenty of conversations around how do we prioritize? Because there are a lot of things that we can do. There are endless things that can be done, but resources are limited. Elli's time is limited, right? And so at a certain point, we need to make a call on where we think we have the highest probability of success and focus on that. As it is, it's challenging. Even to take that one therapy through is challenging, right? And so we decided to go for the root cause that is specific to FOXG1. We know that the vast majority of FOXG1 syndrome patients have a haploinsufficiency. They have 2 less of the activity, and supplementing it with a gene therapy is a direct mechanistic solution to try and improve on symptoms and overall quality of life. And that's the path that we picked. Is that the only path? No. Could there be other ways? Yes. Are we going to explore other ways? In a way, we are already, but we need to prioritize our efforts and resources, and we're going for what we believe is the strongest mechanism of action to treat.
Yeah, and when we decided that, we spent a lot of time with the Angelman and Rett syndrome communities, who they're bigger, and they did spend quite a lot of money on drug repurposing, and none of those efforts were successful. And so we learned a lot from what others had done. And as Guy mentioned, it's not that we wouldn't look at something like that in the future, but it's right now we're prioritizing on the genetic therapies.
Tim, do you want to push back?
All right, here we go.
Nasha, we got.
Oh, do you want a mic? Is there?
Can you hear me? Working in the Rett space where the outcome measures leave a lot to be desired, and what happened is that with subsequent trials, the FDA basically said, well, we want to see the outcome measures that you used in the previous trial. But the scientific community was saying, but we hated those measures all along. So to be doing what you're doing right now is to establish measures that work, that measure things that are important to all of you. You're setting the groundwork for not just a gene replacement therapy trial, but any sort of drug that might come along. Because some people may not be able to participate for whatever reason in gene replacement therapy, but you want them to have something. But if you have the outcome measures, you can then do that trial and decide, okay, that works. Because what may end up happening is it may not be gene replacement therapy that leads us to where we want to be, but it's that plus something else plus something else. And having the measures that can measure this accurately and appropriately with things that are meaningful to you is huge. So to your point about everybody participating, just to reiterate that, it's huge.
Thank you, Doctor.
Thanks.
Thank you.
It's a
Thank you,
commercial.
Tim.
Sorry.
Yep.
Yeah, thank
No,
you.
this is spot on. Thank you so much.
Ryan, I
Not
think
sure.
I'm going to have to
We
call
have.
it.
I think there's one more minute
Okay,
here
all
if
right,
we
all right.
can, and I think it's an important one. Sorry, just fitting it in.
Very hungry.
Has cure been defined? How do we define cure? What does it mean? And what is the actual realistic goal and outcome of this drug? I think that's important for
Yeah,
our
it's an
community.
important question because cure is a loaded word. Do one of you want to take that?
So I think that in a way this once again ties to what Terry said earlier about a possible sliding scale of treatment effect as it relates to either age or genotype or both, etc. And I think that cure is it's a complicated word because it can mean different things to different people, and I'm not sure how to define. I haven't checked Merriam-Webster, so I don't know how cure is defined in the dictionary. But when I think about what we're doing, I view it as treatment that really changes fundamentally the lives and quality of lives of patients and families, and gives functionality and quality. And I don't know how it's easy for me to define treatment because it's tangible, it's functional. We know what we're aiming for. Cure is aspirational. It's a word that I just don't know how to anchor in a clinical paradigm, because in a way it at least to me suggests that we're like miraculously sort of making the condition completely disappear, and everybody bounces to be like completely 100% neurotypical. And I hope that this is achievable, but I don't know that it is. And even if it isn't achievable, it doesn't mean that we can't have a profound effect on the overall quality of life of everybody.
Yeah, and you actually just took the words right out of my mouth, that I think some of the framing around this is related to quality of life. And we were talking about this at dinner last night, but if you had asked our team maybe like 5, 6, 7 years ago what that meant for us, I think we probably would have said something to the effect of, even modest changes can significantly improve the quality of life for the patient and the family and the caregivers and the network and system. But some of what we're seeing coming out of the gene therapy trials that are active today, other modulators of gene expression, have been beyond, I think, what any of us might have imagined before. And so I think there's reason to be quite hopeful as well that we will see transformative change.
Yeah, and I think every family will have to make that decision when you decide to enroll your child in a clinical trial or even give your child the approved drug. What does it mean for you, right? For me, having Amara say a couple words, be able to help herself. So my biggest fear is when I pass away, can she call for help if someone's abusing her? And that's what I think about. And to me, that would be worth it. But every family has to make that decision for themselves. Hopefully that helps.